Okay, so last lecture in this chapter is on the extracellular matrix, and this will be the last of 4 different structures at the cell level that we are basically learning a little bit more about so that we can start putting them together in the material that follows. So the extracellular matrix in many ways is very similar to the cytoskeleton, except it's on the outside. And so, as the term extracellular suggests, it is outside the cell, and it is a matrix in that it is this crisscross network, and also other components with the fluid outside as well. And it's important for you guys to realize that while obviously we've been talking about how cells come together and make tissues, you do need to realize the tissues are not just the cells. The cells also are going to make and secrete things around them to essentially create for themselves a home, an environment in which they are going to live. And that's going to have some solid components to it, that's going to have some liquid components to it, or fluid components, and the kind of extracellular matrix we build is going to be determined by the composition and architecture that we need as a tissue type. And hopefully that'll kind of make sense as we go through some of the examples. So primarily this is going to provide a way or a place in which cells can live, they can move, they can grow, they can interact with each other, and they will also be able to interact with it. The cells in a multicellular organism for sure are going to build some of these things. If you think about plants, the cell wall would be a very good example of an extracellular matrix. In our case, our extracellular matrices are much less rigid and structural than a cell wall would be, but they are still pretty important for those types of roles. So typically when you look at an extracellular matrix and what goes into it, it actually has 2 components to it. One is a network of fibers, and there are several different kinds that may go into them. And similar to the this would be the intermediate filaments of the cytoskeleton, the fibers in an extracellular matrix are going to prevent stretching or tearing, and then the filler materials are going to resist the compression. So it's almost like the filler materials are going to be almost like Jello, where if you want to keep something safe, or if you think about the sometimes you'll get a box in the mail and it'll have whatever it is, like a camera or whatever, it'll have the foam that's basically been sprayed all the way around it, and the foam can be destroyed, but in and of itself it will resist compression so that it protects the camera inside. So these filler materials inside the extracellular matrices are going to do the same thing, where it protects the cells that are living inside of it. So whatever the form, and we're not going to really get into a whole lot of details on this later in the course when we talk about specific kinds of tissues like kidneys and livers and things like that. We may throw a little bit about this out there, but generally the proteins tend to have similar characteristics. They usually are fairly elastic and fairly stretchy. Again, we're trying to keep you from being torn apart, but we don't want to resist. Basically, we want to resist the tearing, but we want to give flexibility. And I'm sure you guys have heard of some of these kinds before: collagen, elastin. So these are things that you'll find written on hand lotions and stuff. And certainly, I guess the idea is that if your hand lotion has collagen in it, then it's going to help with the extracellular matrix of your skin and make it more elasticy. The filler components are what are called proteoglycans. These are massive structures, some of the biggest molecules that we have inside of our bodies. And as you can tell, they're somewhat of a combination between proteins and sugars. And again, it kind of makes this almost Jello-like substance that fills in the space around the extracellular matrix. And of course, in and around these things would also be what we call the interstitial fluid or extracellular fluid. And of course, this is what's going to exchange materials with the cytosol that we've talked around before. Whenever it's time to transport sugar inside the cell, it'll go from this fluid to the cytosol across the plasma membrane. This would be the contact points with the plasma membrane. So here's a somewhat cartoonish diagram. I couldn't really find one that was better than this that had things labeled, but you really can see some of the things in here. So long collagen fibers, and these little molecules that connect them, laminins, and these would be the proteoglycans. Okay, actually this would be the proteoglycans here. This right here is another one you might have heard of. It's called hyaluronic acid, and that's what your proteoglycans are attached to. So that's kind of the filler, and then these fibers crisscross throughout to give us the other piece. And then there's going to be connections with, we'll get to these a little bit later, these integral proteins in the membrane called integrins that are going to be able to connect us to the extracellular matrix that we're building. So all of these components would come from the cells and are built by the cells and secreted and will be assembled outside. I really like this picture because it shows some of those components. Here's your proteoglycans hanging off your hyaluronic acid, and here would be some cells living in the extracellular matrix that they've made. And I like this picture too. I threw it in here because it really shows you the purpose of both pieces. So the fibers are in there to give it strength. So this is what, in a case of a bridge or a building, would keep the building from ripping to shreds, whereas it's the compression material that allows cars to drive on it or people to walk on it because of the downward force. The concrete protects from those forces. And when it was discovered, the matrix was generally believed, it was outside the cells, so it was generally believed to just be something that the cells made and then left it alone. But since then, it's really been discovered that the extracellular matrix is just as dynamic as any other part of the cell inside. And sure, cells are the ones that are having to manage it, so they will digest away pieces and recycle pieces and secrete things to replace them. But the bottom line is that the matrix can actually be changed, not so much in real time, but in response to things that are going around the cell. So one of the reasons why we will see it is that the matrix plays some roles in behavior, essentially how cells are going to act and what they're going to do. It's going to play a big role in helping cells migrate, because just like the cytoskeleton provided a mechanism for organelles to walk around and get to where they need to be, the extracellular matrix provides something that cells themselves can walk around on. And we'll see that the extracellular matrix we make is very important for development. And by development there, I mean taking on the role that you're supposed to take on. And of course, cell division can also be affected by the nature of the matrix. And matrix can kind of affect where the division takes place. Is it going to go this way? Is it going to go that way? And of course, those relationships with regards to those axes can be important. The function of the extracellular matrix, depending on the tissue it's in, will be determined by what it has in it. And so I just want to give you a few examples here. There are some that are packed so tightly that you would call them a basal lamina. It's basically an underlayment for other tissues to build on top of. Very strong, very hard to break. And then you may also have others that are a little bit looser. Some that are going to have the fibers but are going to absorb water, so that they're very spongy. So for example, the basement membrane of something like skin would be very stretchy. We can't tear it very easily. You'd have to put a lot of force on it to tear it, and that would be because of a basal lamina underneath the skin. Cartilage, as you know, is very spongy, so very high water content in there with the cartilage fibers. Bone, of course, is very dense, very brittle. The extracellular matrix would be calcified, So the calcium would help harden it. Tendons would be very tough. If you think about getting a tendon with, if you get a tendon or a piece of gristle in your steak, very hard to chew through that. And it's basically those kinds of extracellular matrices that are going to be the strength that makes a muscle attached to a bone and actually be able to move that bone around without actually ripping loose or tearing. So here's a couple of pictures. So here you see a loose extracellular matrix with the collagen fibers, and I think this would be a picture of skin here. Here would be your basal lamina, or your basement membrane, it's called. So very thick and very tough. And then of course the layers of cells on top of that as they create the skin. So this would be near the dermis layer, and then it would create the epidermis above it. This one, so this will definitely be a role that's played during development. So you remember we looked at what blastulas look like with a hollow ball of cells early in the developmental process. So this green is a fibronectin extracellular matrix, and what happens is, so this starts out as a hollow ball of cells, and as the so this is going back in time a little bit, what would happen is we would make a fibronectin layer, and then what happens, oops, thought I could erase just a little bit of that, but what will happen is that the cells are going to move inside and begin to crawl along that surface. So what you're looking at in that picture is, this is going to be the gastrointestinal tube, and this will either become the anus or the mouth, depending on what kind of animal we're talking about. But what's happened is this used to be a hollow ball, and then some of the cells dipped inside and began to crawl along that inside of the fibronectin, and are going to pull it all the way to the other side until the tube goes all the way across. Pretty neat. I think I got one more picture here. Let's see. Oh yeah, this was kind of neat. So basically what this figure shows is, slowly over time, about a 12-hour period, these researchers, so this was in a deli box, a fridge, and what they did was they slowly dripped a saline solution, and one that had components in it that would actually remove the cells from their environment. And so the blood cells, the muscle cells, and other things basically get removed as they slowly drip out over time. And what you can see here is that once all of the cells have been removed, you still see the organ. And so what this tells you, I think, is, or the neat thing, the story this is telling, is that the organs, these shapes that you guys learn the different organs. So you remember those models that are in an anatomy lab, the liver and the small intestine and so on, and it goes here and you're putting them together. It's actually the extracellular matrix of each one of those organs that is created by the cells in that organ to actually give you that organ, that shape. So it's not the cells that give you the shape. Well, I mean, it is, except they're actually building that shape as an extracellular matrix so that they can live in it. And so a liver is shaped like a liver because the cells make that extracellular matrix, and that's what they want to live in. And a bone looks like a bone. So like after you've eaten a chicken leg, you would have that bone. That is an extracellular matrix, and eventually all the cells would have been removed from that, and the bone is still left behind as the extracellular matrix. So it's just kind of a neat concept there to think of all the shapes that we learn about in the anatomy of the body are really there because the cells that live in them built them that way. Okay, alright, so that finishes off chapter 2, and we'll get into chapter 3 and actually start tying some of this material together. One, we'll do some genetic stuff, and then 2, we'll do some getting cells together into larger structures over the next several chapters. All right, so see you then.